Medicated weight loss is not cheap.
Hey, hi guys, welcome if you are joining me. I am currently running 2 sessions at the same time, so I'm hoping that this will work for everybody. So if you are with me here, then welcome. If you are just joining me, also welcome. Today's session, what I want to do is I would like to look at photosynthesis. So we started photosynthesis last week and I want to continue with photosynthesis. But what we're also going to do is I'm going to bring in a little bit of the content to do with the topic of organization, enzymes, and the digestive system. So if you are watching on TikTok, I hope this is working. If you are obviously on my YouTube channel, then you'll be able to see this diagram, but I am hoping that this will be showing you the screen. Please feel free to write comments and ask questions as we go. I will keep answering as we go. If you have any questions, so please do ask away. But let's get started. So one of the required practicals that you have to know about when it comes to biology for GCSE is required practical activity 5. So investigate the effects of light intensity. So I spoke about photosynthesis last week, and you should know the equation where you have your water and your carbon dioxide. Light is absorbed by the chlorophyll molecules, and that light energy is used for the reaction, which is going to produce our glucose and our oxygen as our waste products. So what we're looking for then is to understand if it's a required practical. They could ask you to design a practical, or what they might do is they might ask you to not just design the practical, but also to pick out some of the different parts which are, for example, the independent variable, the dependent variable, and so forth. So on this one, it's the effect of light intensity. So we need to think about, well, our light intensity would be our independent variable. So we would move our lamp distance. Now we would start at a distance of 0 and then maybe 10, 20, 30, 40 and 50 millimeters. So if you are going to be giving them this information in a question that says describe how you will change the light intensity, make sure you give them 5 values. So that's our independent variable. Then what we need to do is we need to think about, well, what is our dependent variable? So our dependent variable, therefore, would be the rate of photosynthesis. So how is that going to be measured? So it could be that you're going to count the number of bubbles. So that's the most simplified way of measuring photosynthesis. If you've done this in class, you'll have stuck a aquatic plant into a sodium hydrogen carbonate solution, and maybe you have counted the number of bubbles. But how big was the bubble? How big was that bubble? So therefore, how much gas did it actually have? So what was its volume? Can you calculate that? Not really. So they may ask you to design it, or they may ask you to improve the investigation. So if it's talking about the rate of photosynthesis, we're looking at our gas collected used in a given time. So how much am I collecting? So is it the volume of the gas that I'm collecting? For example, if I'm looking at photosynthesis, I'm looking at the volume of oxygen that is being produced If I'm looking at the volume of carbon dioxide that's been taken in, that is different. So do make sure that you read the question carefully as to what they want you to measure. And then with every investigation, what you need to do is you need to note what are your control variables. So if I am controlling, for example, my if I'm changing, should I say, my light intensity, and I'm measuring my rate of photosynthesis through the gas of oxygen that has been collected, then I need to control my concentration of carbon dioxide. Guys, thank you if you are watching this. I'm hoping it's working both on TikTok, displaying my screen, as well as it does on my YouTube channel. So if you can let me know on both, if you can see the screen, that is great. But going back here, control variables. You should have a list of variables that you want to look at and whether or not they match the investigation that you're actually looking at. In this case, photosynthesis. So I must control my concentration of carbon dioxide, the volume of water that has been provided, the size of my pond weed, the number of leaves that they have, or the temperature. So I would want to ideally have this in a water bath. Well, that would be the better way of doing it. If I have it in a water bath, that's therefore going to ensure that I am controlling the temperature. Why do I need to control the temperature? Why do I need that? Well, if I don't control the temperature, then that could have an effect on the rate of photosynthesis. And I'm going to look at how these interlink, but you've got to remember they're assessing your skills, okay? Guys, thank you for joining me. If you are on my YouTube channel or on my TikTok, please do feel free to ask questions as I go. I am doing this live and I will obviously try and answer any questions that you have. So please do ask away as I go. Okay, what type of question could they do? Well, a lot of students have done the required practical in lesson and they have measured the rate of photosynthesis, they counted the bubbles, and then they throw a spanner at the works at you in the exam and they say, ooh, this person investigated the effects of different colours of light. So in this case, because we are changing our different colours of light, instead of us changing our distance from the light source, we are changing it and it's going to be at room temperature. They've told us in the question. So it's really important as you're going through a stem of a question, which is what we would call this, that you do try to ensure that you've pulled out of the question the hints that they give you. Now, whether you're GCSE, whether you're A-level, the exam boards will always give you little hints in the question. So do make sure as you're going through that you are using the information that they've given you to figure out what they actually want. So then it goes on to say, okay, they've used a pondweed that's in water. The piece of pondweed was placed in the red light. So they've just told us it's a piece of pondweed. They haven't told us how big is that pondweed. They've said it's placed in red light, then they placed it in blue light, and then they placed it in green light. Now each color of light was the same intensity. So they have given us a bit of information here because they've said the same intensity. And then they want us to describe how the student should make accurate measurements to obtain valid results. So it's really important. We're looking at these accurate measurements and these valid results. So what is it that we want to talk about? Well, remember what I said before. They could ask you about your independent variable. They could ask you about your dependent variable. They could ask you about your control variable. So make sure that you have a way of identifying to the examiner, my independent variable is what I'm changing, the colors of light. My dependent variable is what I'm going to measure. Now, my dependent variable in this case is going to be the rate of photosynthesis. Now, you need to ensure whenever you give your answer that you are talking about that rate, and that rate would have to be the quantity that you are collecting. So the quantity that you collect in this case could be my oxygen, not concentration, let's try that again, my oxygen volume. But because I'm talking about rate, do not get caught out by not giving a time. The quantity that's collected, the volume of oxygen in 1 minute. You'd be surprised how many students lose that 6th mark because they haven't been specific enough. So the rate should always be quantity over time. The other thing you have to note as well is that it's asking about valid results, so validity. What are you doing to ensure that you are getting a valid set of results? So have you done repeats? Always with any investigation, you want to ensure that you are repeating the investigation so that you can calculate the mean and standard deviation. Now, for those of you who are just joining me and have not worked with me before, I have been teaching for over 20 years and I tend to specialize with all OCR A-level. However, what I'm realizing is for the majority of A-level students, they are missing these vital points which are GCSE level. So I've decided to help you guys out by doing some more GCSE for you, and this will be very valid when it comes to your A-level. If you decide to do biology later on, you will need to say these same things. You will need to identify your independent variable, your dependent variable, your control variables, and then you want to say, well, if I increase validity, I need to make sure I've repeated, and I'll calculate my mean and standard deviation. Now, for you in GCSE, you don't need to know that word standard deviation, but you do want to talk about, we're looking at any variability in my data. So what we want is to identify if there's any variability. Excuse me whilst I do this, because I'm doing this live. Variability in my data. And one of the things I say each time I do this, I always say to you, I know my weaknesses. And I am exposing myself here by doing this live and talking to you as I do it. And the reason I do that is for you to know I have been doing this for over 20 years and I still make mistakes in my writing, in some of the things that I say. It's okay to make mistakes. However, I am aware of that. So I slow down, and what I will then do is I will make sure that I am taking my time, especially when I am writing things in an exam. Do you do that? Do you slow yourself down? Do you double check your answers? Because if you don't, that could be something that's holding you back. So identifying your weaknesses is a really strong thing to be doing as you're moving through. Okay, so what else do we need to talk about then? So we talked about the rate. We've talked about our independent variable. We are going to give them some control variables. Now, they haven't controlled the piece, but I would say they should have controlled that piece of pondweed. So I would want to give them information saying things like the size, the number of leaves, for example, the concentration of carbon dioxide. So if I am measuring the volume of the oxygen, I need to ensure that I have given them the correct concentration of carbon dioxide. Otherwise, that may affect my results. So we always want to be giving as much information as we can. But what they love to do is throw a different example at you. And they love to give you something that looks like this. So we've got light intensity is going to vary during 24 hours. The overall exchange of gases for a tree also vary at different light intensities. So they may give you a diagram. This is my little diagram that I've made today. They may give you a diagram and then say to you, okay, I can show you that in bright light, we've got our carbon dioxide, and I've got an arrow representing where the carbon dioxide is being taken in and where the oxygen is being released. I've got dim light, but I've got no carbon dioxide and oxygen, no arrows, and I've got no light. And on that one, I've got oxygen, but it's going in, and carbon dioxide that's going out. So if they give you a question that's like this, what they want you to do is use the diagrams. They say that in the question. So I've adapted a question from an exam board to show this. Using the diagrams, you'd be surprised as well how many of you get questions like this and who do not actually use the diagrams. You just start going, I'm going to blurt on paper. I'm going to tell you what I know. That's not what they want you to do. They want you to use the diagrams to explain the overall exchange of these two gases. So can you explain why there is carbon dioxide going in, why there is oxygen coming out? So we're talking about the overall exchange here. So if we're talking about the overall exchange, what have we got when it comes to our first one? So if we've got bright light, so what do we know about bright light? Well, we know that we're looking at photosynthesis because that's what I've been talking about, right? So I've been talking about photosynthesis. If I can spell photosynthesis, that would help even better. So photosynthesis. So again, I'm slowing myself down. Thank you. Photosynthesis. Ah, Hannah, you've jumped in here. Nice. And you've said respiration, but we've not been talking about respiration right now. So we're not talking about respiration. I'm talking about photosynthesis, but you said respiration. Does that mean you're wrong? No, it doesn't. Okay. It does not mean you're wrong. It means they want you to bring in both. They're checking if you know that photosynthesis is in one direction and respiration is in the other. So if I start with photosynthesis and I say, okay, I've got water, I've got carbon dioxide, and when I have water and carbon dioxide, I will then make my glucose and my oxygen. And remember, your sixes, if you're a higher level student, you must be able to balance that equation. And you're absolutely right. Respiration is always happening, whether it's an animal or whether it's a plant. Those cells will require energy, the energy to do metabolic processes, and therefore they are going to require glucose and oxygen. So if we know that respiration goes in the opposite direction, we know that we've got glucose and oxygen are going to come together, and that therefore will produce water and carbon dioxide as a waste product. We should know that these two are the reactants for respiration, whereas these two are the reactants for photosynthesis. So whether they are the products or reactants is really important when we are looking at the equation that we have been given or the question that we're being asked. So what happens in bright light? Well, we know that respiration is always happening. So that makes sense. Oxygen is always going to be required if it's aerobic respiration. And we've got the glucose, and we will therefore be making water and carbon dioxide. So why is it that the carbon dioxide is going in and the oxygen is coming out? If we're always respiring, why is it that that's the way that the arrows are going? And that's what they want you to tell them. Yes, we know our photosynthesis equation. Yes, we know our respiration. EM, you're saying the rate of photosynthesis increases. What do you mean by it increases? Why? Why is it increasing? What is it that makes it increase? And this is the important part. They have told us that we are in bright light. And we know that if we have a plant that has chlorophyll, we know that that chlorophyll is going to be able to absorb the light energy. So if we have chlorophyll and we have a bright light, we know that that is going to be absorbed by the chlorophyll molecule. The carbon dioxide and the water will be able to be reacted together to produce oxygen. So because we're in bright light, that therefore means that there is extra carbon dioxide being absorbed from the atmosphere, And therefore, the rate of photosynthesis is greater than the rate of respiration. If more carbon dioxide is coming in than going out, that therefore means that the carbon dioxide coming in outweighs that that's going out. We can be sure that there is a greater rate of photosynthesis compared to that of respiration. So the oxygen is being produced through the process of photosynthesis is therefore in excess of that is needed for respiration. And because of that, that's therefore meaning it is being lost to the environment. It is being exchanged. It is leaving the plant. So if you are looking at your leaf structure, you should know and you should remember that you have got your stomata, which are surrounded by your guard cells. And when they are open, they will allow for the carbon dioxide to come in, and they will allow for the oxygen to diffuse out. They will allow for that gas exchange. And that gas exchange can occur only if there is a concentration gradient. So if oxygen is being used in respiration, there won't be excess oxygen to leave through the stomata. So it's important to note that when you are looking at biology, and if you're aiming for that high grade, if you're aiming up towards the 9, you should know keywords like the stomata, the guard cell, the fact that this is gas exchange that's occurring. So when we look at the bright light, we've got this rate of photosynthesis being high, extra carbon dioxide being absorbed, oxygen is released into the air. So what is causing that? Where does it come from? What is going to happen? So make sure as you're going through that you are checking that you know your keywords, that you know how to extend on just what's been given in the exam. So this is where a lot of students actually lose out because they think, well, I've said enough when I talked about photosynthesis is happening, but it's this comparison that will make the difference. So what's happening in dim light then? We have no arrows going in or out. Does that mean there's no gases moving? Well, no, it's not that there's no gases moving. The light is dim. Photosynthesis is still occurring. However, remember when we talked about those equations, let's just bring that back down again. Here we go. We've got our equation back again. So if we've got our equation back here then, we know that photosynthesis and respiration are happening. Yes, Hannah. So the rate of, hi Tom, thank you for joining on this channel too. The rate of respiration and the rate of photosynthesis are now equal. So the carbon dioxide that's coming in and being used in photosynthesis is equal to the amount of carbon dioxide that is actually being produced through the process of respiration. So because they are equal, it means that there's no extra coming in and no extra leaving because it is no overall exchange. And so that's, yes, I am live, Tom. I am absolutely live right now. Thank you for writing this. And I hopefully I'm still being shown on both both platforms. I'm doing this for the first time where I'm actually doing it on TikTok as well as on my YouTube channel. So it should hopefully, I'm assuming because you guys are writing answers that you can all see it on the TikTok side of things. But if you can't, I am displaying it on my YouTube channel as well. I've been trying to get it to work for a while now, and this is the first time I've done both, so do bear with me. And I'm freezing again, and my face is so warm. My hands are like so cold. It's crazy, but never mind. Right, carrying on. Last one then, no light. So when there's no light, well, obviously, if there's no light, are they photosynthesizing? That's the first thing to think about. What was the important part? It was light. Light is needed for photosynthesis. Because light isn't available, is the plant photosynthesizing? No, it's not. So if it's not photosynthesizing, what is it doing? Well, it's still respiring, because we know respiration occurs all the time. So one of the things for you to note here is, and I've left these indicative marks on there for you. Let me just bring that up again. Whenever you're asked a question like the one, so if I bring the question back down, which was the original 6 marker. So the original 6 marker was this. There we go. Let's just move it up here. So there's my original 6 marker. Using the diagrams below, explain the overall exchange of carbon dioxide and oxygen at 3 different light intensities. So as an examiner, when it's a 6 marker, what I would be doing is I would be looking, in fact, let me bring the first picture down and show you exactly how I would do it. So I've just shown you all the indicative marks. Yes, so Tom, interesting, Tom's just said that he was learning this for his A-level exam. And yes, I am on both, Hannah. I'm live on both sides. So what I would say to you is, this is the reason why I'm doing these free lives on Monday, because what I'm realizing is so many students who are doing the A-level don't actually understand that it is a step up from your GCSE. And if you don't have that foundational knowledge at GCSE, the A-level becomes much more challenging. So I want these videos to be available for GCSE students who are sitting their exams this summer, but also for those of you who follow me through with your A-level, so that you can go back and refresh your knowledge before moving into that really hard concept of light dependent reaction, light independent reaction, non-cyclic, cyclic photophosphorylation. We go into way more detail at A-level, but the starting blocks are here at GCSE. So let's nail that content so that when you get to me next year, you're already in a better footing. And that's the hardest thing I would say for students, is that they cram up to the exam for their GCSE. They get to the summer, they've kind of been spoon-fed the information. They go, they relax, they get into A-level and go, what? You want me to know it and more, and you want me to learn this and be able to apply it. So this is why I'm doing this. And if you are at Tom A-level, I will be back at 9 o'clock after I've done my group session to do free live for A-levels as well. So if you do want, come back and join me later on. Anyway, back to this then. So I have my tray. Photosynthesis has occurred and we have got glucose. Now we're going very basic, obviously, because we're at GCSE level. So we have got glucose as our chemical energy because we've got our molecule. Now, what's going to happen to that glucose? Well, that glucose could be used in lots of different ways. And again, if you are an A-level student, you're literally going to go like this and be like, hold on, that's exactly the same as what I need to know for my A-level. And it is. It's very much the same, but just with a bit more. Oh, you just found my channel. Wow. Well, I'm glad you found it. Now you know who I am. If you're doing OCRA, the whole course is on there for you on my channel. So you're welcome. If you're doing AQA, 75% of it is overlapping. So do still check it out, but just double check it against your specification because they don't do, for example, viruses in more detail. Right, glucose. What can we use with the glucose? The glucose could be used to make cellulose. That cellulose is needed for building the cell walls. So when the cells are going through absolutely growth, and when that cell is needing to go through the point in which mitosis has occurred and that has then resulted in 2 genetically identical cells, what we've then got is the building of the cell wall. So when the cell splits apart through the process of cytokinesis, that cell wall has to be formed. Well, the cell wall, we know we can distinguish plant cells because they have a cellulose cell wall. Well, that came from the glucose that was an end product of photosynthesis. So that's one way in which the plant can use glucose. What it can also do is it can use the glucose and store it as starch. And if it stores it as starch, a polysaccharides, so biological molecules coming in here that you go into more detail at A level. What else do you need to know? Well, how do we test for starch? Again, very similar. Iodine. So when you're at GCSE, you need to know it as the iodine test. A level, you need to know it's the iodide and potassium iodine and potassium iodide that are put together to give you the iodine test. Same content, just in more detail. Same results. Negative results, orange and brown. Positive results, a blue-black color. So if we're looking at what happens to the glucose, what is happening as the plant is growing, either it's growing, making more cells, using it for cellulose. It could have been respiring, obviously, we've already talked about that glucose being used for respiration, therefore it's no longer available, or the glucose has been converted into starch. If it's been converted into starch, it's a storage. But right now, it is winter. It is cold. The leaves have fallen. Because the leaves have fallen, the plants still need a source of glucose. So by storing it, either within the cell or in storage organs, means it's available when there's less light for photosynthesis. Now, the other thing it could do is it could be converted into lipids. So if it's converted into lipids, what would we then do with it? Well, again, looking at the emulsion test, we can look at a way of linking our topics together here. And this is the thing I love about biology, is the bigger picture, seeing it all together. The glucose gets converted into the lipids, the oils. For example, I love a bit of olive oil when I'm cooking, or if I'm going to have it on toast with some avocado. Well, what am I doing? I'm using the plant's oils in my cooking. So that's when, if you're doing chemistry, that needs to come in, obviously, with why would you use those kinds of materials? Anyway, let's come back to biology. If we're going to do, the glucose is used for the lipids, and we want to test a food source to see if there are lipids present, we would do the emulsion test where we use ethanol. Positive result, we'd get a white precipitate forming. So the way it reacts, it shows us this white precipitate. That therefore means we can say, yay, we've got a positive result. If it were negative, it would be a clear solution. So always, again, if you are revising your GCSE, do make sure that you know not only the positive results, but also the negative results. Because if they ask you about your food tests and they ask you to describe what you would do, you need to give them that note of it would be a negative result if it were clear. It would be a positive result if it went to a white precipitate. So really important, you know both the positive and the negatives. What else can happen to the glucose? Well, the glucose could actually be combined with nitrate ions and therefore be used to make amino acids. So those nitrate ions that were in the soil, those nitrate ions which are actively transported into the root hair cells, those nitrate ions which can be moved through the plant with the water, can then be used to take the glucose and convert it into amino acids. And what do amino acids make? Well, if we join them together, they're going to make proteins. Well, how do we test if it's got proteins? We do the Biuret test. What's the negative result? The negative result is blue. The positive result is purple or lilac. I always say proteins, using my alphabet, proteins. Proteins are purple. So do make sure you have your little cheat sheets. And that's what I try to make when I do these diagrams. I did this one this morning for you because I knew I wanted to help make it visual. I am a visual person. I am someone who, if I am trying to do any type of revising, I would have a diagram like this and I would have it going off in all the different directions. And then what I would do, I would then later on, after I've done a nice perfect little diagram, I would then close my eyes and I would test myself in each of the different parts of my sheet. Where were those different key bits of knowledge. And the reason I do that is it's a really good way of priming your brain and helping you to remember what you need to for the exam. When you're sitting in that exam hall, and this is another little trick I will teach you that I've learned a number of years as being a director of student progress and head of biology for my students. If you understand how to make your brain work for you, you will be more successful in that exam. So what I'm going to say to you is, find out where you're sitting your exam. Find out if it's in the main hall, find out if it's in the small cafeteria, depending on how your school is set up. Find out where your exam is going to be for each of the different exams. Now, in your bedroom or your study room, wherever it is that you are doing your studying, have it that one wall has got one set of notes. One wall has got another set of notes. One wall has got another set of notes and another set of notes. So if you've got 4 walls, obviously your room will just be covered in them. And that's what you want it to be. So that when you are looking at this, you can then, in the exam, if I'm facing forward, I know that when I'm sitting at my desk, I'm looking forward. That's my notes on photosynthesis. Oh, hold on a minute. This question's asking me about respiration. My notes are over here. Adam, yes, I am going to do A-level today at 9 o'clock. So I'll be on later on again after I've done my group tuition session. So please do join me if you're looking for A-level. But back to this. So if I have that my specific notes are here, then I know that that's where I need to try and picture it. And I get my students to actually think about the fact that if I asked you, and we do this in the lesson quite often, if I asked you to close your eyes and make me a sandwich, and I asked you to go to the fridge and I asked you to make the sandwich and bring it to me, what I actually get the students to do is talk me from getting from a room like the living room all the way to the kitchen, getting the sandwich out, making the sandwich, and then bringing it back to me. Now, as they're doing that, they do that every day, all the time. They're exposed to it and they don't think about it. But the minute I make them think about it and they have to visualize it, you see them close their eyes and then they're like, well, I walk out the room and then I turn it and they give me all the directions. That's what you want to do. You want to see it that every time you go into your room, you're being exposed to this content, but you don't realize what it is. That's basically what TikTok, what YouTube, what this is all about. We actually, as teachers who are on here, we're trying to help you because we know social media is amazing for keeping you occupied, but unfortunately it's so easy to scroll for 4 hours and be like, oh, I didn't do any revision. Whereas hopefully my video pops up and you go, oh, how did I manage to stay here for like 40 minutes? So one thing I would say to you is have a place in your room so that when you're in that exam hall, you can then say, right, this is where my notes are. I've got a trigger into that information. Tom, back to you. Unpopular opinion, but I think AQA is much more harder than A levels because of the level of complications. AQA is not more complicated. I would actually disagree with you on that one. Each of the exam boards have all got level 3 qualifications. Okay, so whether you're doing AQA, whether you're doing OCR, whether you're doing EdXLB, whether you're doing CIE, all of them are level 3 qualifications. So GCSE is level 2, A level, IB is level 3 qualification. And it doesn't matter which exam board that you do, they have different styles, but they also have different amounts of content. So I actually would disagree with you. Now, majority of students do actually sit AQA, so therefore you get more exposure from the number of students who sit AQA than you do to those who sit OCR or EdXL, B, etc. So it's not that it's harder. They all have their same challenges. It's just about how they assess and what they assess. Because I do a lot of tutoring for AQA and OCR, obviously. I built my channel on OCR, but I do AQA, so I know both of them quite well. And so I would say neither of them are harder than the other. They're just assessed in a slightly different way. And the same with the GCSE. I'd say that AQA is a nicer specification for you to look and see what they want you to get. OCR is a little bit more interpretive as to the detail you need to go to. So that would be my little caveat on that one. Okay, I want to keep going because I've got probably about another 15 minutes that I can do for you guys. So limiting factors. Now limiting factors, obviously, limiting factor is going to reduce the rate of, whether that's photosynthesis or any chemical reaction, a limiting factor is going to reduce the rate of that reaction. In this case, we're looking at photosynthesis. So if I have a graph, and they love either to get you to draw a graph or they love to get you to interpret a graph. So here I have down at the bottom where there is 0 light intensity. So if I have no light intensity and I then increase my light intensity, so if I am increasing my light intensity, what I have got is anything that is before the line plateaus. So in each case, all the way up to the point at which it then plateaus, whatever is at the bottom was limiting the rate of, in this case, photosynthesis. So obviously the light energy is required to be absorbed by the chlorophyll molecules for photosynthesis to occur. So if I am limiting the light intensity, my light source is further away from my plant, that is going to affect the rate of photosynthesis. Another little trick for you, if you are given a graph in the exam and you are asked to describe it, say what you see, give numbers between 0 and 100 lux, between whatever they give you, 20 arbitrary units, whatever those numbers are, actually use them from the graph and give 2 sets of data to back up whatever it is you're saying, especially if you're aiming up for that 9 grades. So whatever is numbers between this point and this point here, I would say between 0 and whatever that number may be, that is limiting the rate of photosynthesis. After that point, it's something else. So if I tell you that this line is low carbon dioxide concentration and low temperature, and this one is a high carbon dioxide concentration and a low temperature. So let's give an example that they maybe say we've got 25, no, let's say 10 degrees C. Let's go for 10 degrees C on this one. This one is 5 degrees C. Therefore, I have, no, that's not what I want to do. Try that again, because that's still low. 5 degrees C on both of them. However, my carbon dioxide concentration, see, doing this live, you make mistakes. Maybe this is 0.04% and this is 0.15%. So if I have a different low temperature, low carbon dioxide concentration, higher carbon dioxide concentration, when the line plateaued off on this one, what was limiting my rate of photosynthesis? It was the carbon dioxide concentration, because that was low, but the temperature was the same. When I look at the next part, as we start to move up, if I'm increasing my carbon dioxide concentration, but I'm plateauing, now temperature is the limiting rate. Because I've increased the carbon dioxide concentration, so carbon dioxide is not limiting my rate anymore. Therefore, the low temperature must be limiting the rate. And if we go up to this one, we've therefore got, if I've got a higher concentration of carbon dioxide, I've got a higher temperature. What is limiting my rate of photosynthesis? It has to be something else. So something else is limiting the rate of photosynthesis. It's not light intensity, because we've plateaued. It's not the carbon dioxide concentration, because we've increased that. It's not the temperature anymore, so something else. It could, for example, be the number of chloroplasts, potentially. So it really just depends on where they want you to go. So it's important that you're looking at the graph that they've given you, and you are able to then use that to help you Now what I'm going to do is I'm going to switch things around slightly. I want to bring in this part here. Let me just bring this in. Because I want to talk about temperature, and I want to finish on temperature, and then I'll go into the digestive system in more detail next week when I do this. So one of the things to note is when you have chemical reactions happening, we know, whether we're talking about biology or chemistry, we know that when we have reactants, when they come together and they collide correctly and they have enough sufficient energy, we call that the activation energy. So you can see the energy that was in the reactants. When they are colliding, they need the right and sufficient amount of energy, and we call that activation energy, and then we will end up with our products. So whether it's an exothermic or endothermic reaction depends on where you'll see your line come off. But it's important to note our activation energy that is required goes from that initial line up to the peak. So that's important for us to know, and we know that biology, we have enzymes that are protein that act as biological catalysts. They speed up the rate of the reaction. If you're aiming for that high grade, if you're aiming up to the 9, then you need to be using terminology that's going to help you. So you need to be talking, don't worry too much about the enzyme substrate complex. However, if your teacher's really good, they should be teaching this to you, priming you for A level. But what you're interested in saying at GCSE is that you have got the enzymes are going to collide successfully, and because they're colliding successfully, it is going to lower the activation energy required to start the reaction. So it's really important that you note that your chemistry for GCSE also, those key words need to come into biology if you're aiming for those high grades. So when the enzyme is going to collide with the substrates, it lowers the activation energy, which makes the reaction happen at a faster rate, which is why we call it a biological catalyst. Because we are lowering that activation energy, we are sending it on a different pathway, making it easier. So why am I talking about this? Well, you should know that enzymes have got this active site that's complementary. You should know that you have different enzymes, and that when they come together, you have that lock and key hypothesis, GCSE level, lock and key. Enzymes catalyze specific reactions because they have a very specific shape, which is their active site shape. That active site shape is specific to the substrates, the reactants that are going to come together. So if you know that, what have we got to do when it comes to temperature? Well, when we talk about the rate of photosynthesis, we know that photosynthesis occurs due to lots of chemical reactions occurring. So if I have 5 degrees C, I am going to have a slow rate of those collisions, therefore slow rate of a reaction. Because there is less thermal energy, there is less kinetic energy, they're therefore going to collide a lot slower, and that is going to result in a slow rate of reaction. If I increase the temperature, so I've increased it, I've doubled it, 5 to 10 degrees C, I've now got a faster collision, therefore a faster rate of reaction. So I've simplified this as much as I can for you at GCSE level. And then obviously 20 degrees C, I didn't even get to take a breath before it came together. If I've got an increase in thermal energy, I am then going to have even more kinetic energy, even more collisions, a faster rate of reaction. So when you look at a graph, when we're talking about rate of reaction and temperature, you always see that graph increasing. It's increasing because as I increase the temperature, I increase the kinetic energy, more frequent collisions, a faster rate of reaction. When I get to what's known as its optimum temperature, wherever that peak may be, that optimum temperature, that is then going to be the fastest rate of the reaction. If I increase past the optimum, I then do what's happened here is I then start talking about denaturation. So many students jump to denaturation whenever they see temperature and they forget about the first part of the graph. What was limiting the rate of photosynthesis in this case? The thing that was limiting it was, remember, what was on the bottom. Until it curves off, that it could then end up with a plateau. What was limiting the rate was the temperature. However, temperature is unique because if I increase the temperature too high, I'm going to increase vibrations. That's therefore going to change the shape of the active site. It is no longer complementary to the substrate. Do not jump up to denaturation when you see temperature without actually talking about what's happening before the optimum. And that's where a lot of students have memorized optimum denaturation but forget about that before. Tom, may I know from where you are doing this live? The US? No, I'm in the UK. I'm from Cambridge. Well, I'm not from Cambridge, but I live in Cambridge and have done since I did my PGC here 20 years ago. Almost 20 years, actually. Almost 20 years ago. So yeah, I'm from Cambridge. I'm doing this live from Cambridge. And I'm originally from Scotland, although I'm not technically. I was born in England. I moved to Germany and then went back to Scotland when I was 6 years of age, because that's where my parents are from. And then I moved to Cambridge when I finished my first degree, and I stayed here, did my postgrad to be a secondary teacher, stayed here to do a second postgrad, and then stayed here to do my master's, and I love it so much I've never left. So I am currently in Cambridge. Right, guys, I am going to stop now because I need to set myself up for my group tuition that's about to start in 9 minutes. Thank you for joining me on here. Thank you for joining me if you're with me on YouTube or TikTok. Thank you so much. I will come back next week. Oh, wow, you're doing your A-levels from Pakistan. That's amazing. That's really cool. I'm glad that... Don't apologize for interrupting. I do these free lives on purpose so I can help people all over the world. I want it to be that just because I am no longer in the classroom physically doesn't mean I can't help support a great number of students. I have the skills, I have the training, and I also want it to be that when my daughters get to the age where they're maybe doing GCSE or they're doing their A-levels, then they can look back at my videos. And if you are watching, because my daughters keep trying to talk to me when I'm on here, because they sometimes watch a little bit. Hi! So yeah, so guys, thank you for joining me. If you are A-level, I will be back at 9 o'clock tonight to do some more A-level content. But please do keep watching, keep sharing, keep liking, and thank you for joining me.